Okay, so I think we can probably begin. So, welcome to Proteonics webinar today. As I start, someone else needs to be admitted. Welcome to the webinar on unleashing the full potential of CHO cells and what is the importance of vector technology. I'm pleased to be able to invite Maurice van der Heijden to talk all about this. Before we do go there, I guess I'll introduce myself. So I'm Jonathan Frampton. I'm the Director of Business Development at Proteonic. If you do have any questions during the webinar, please feel free to click on the Q&A and then post a question. You can post it anonymously, or you can keep your name depending on your preference. And then at the end of the webinar, we will try and get to all the questions. If we don't manage to get to your question, we will get back to you afterwards as well. We're hoping to get a recording of the webinar out in next week or so. So if you do have colleagues that you feel could benefit from seeing the webinar, then you can email me or Maurice, and we'll be able to send and share a link to you. Okay, without further ado, let me hand over to Maurice. Over to you, Maurice.
Yeah, thank you, Jonathan. Yeah, welcome to the webinar. My name is Maurice van der Heijden, Director of R&D at Proteonic. Yeah, so do you get the most out of your CHO cell platform, or is there still room for improvement on the vector side? In the next 30 or so minutes, I would like to show you that the plasmid vector can be sometimes overlooked, but very important factor with significant impacts on the performance of your CHO platform. So in this presentation, I'll give a short, some general criteria for good expression vectors. I'll explain what 2G Unic technology is and how it's been used in making well-performing cell lines, so show some real use cases. But let me start by introducing Proteonic in a single slide. So Proteonic is a Dutch biotech company founded in 2004 with labs and offices in Leiden in the Netherlands. We're established around 2G Unic technology, well, basically vector technology for enhancing protein expression, transcription and translation. It's functional across various commercial production platforms, and it's not completely limited to mammalian systems. So we've also developed variants for instance, for yeast and pichia, but today's presentation will focus on mammalian cells. It's patented. We have a solid track record and client base with 2G Unic cell lines in clinical development, being used by top 20 pharma companies and in CDMO partnerships, and we commercialize technology by licensing. So, and below are a number of companies that are using the technology that took licenses since we started using the technology, since we started marketing it in 2018. So over to the technology. So if you have a cell line and you want to improve the yields, there are many things you need to take care of. You need to choose a medium, optimize the selection procedure, optimize your process for expression of your cell line, and you need to optimize the use of a vector. So at the performance, you need to optimize all these things because the performance is really dependent on the complete platform on all these factors. And the plasmid vector is one of the major factors in this for the expression. So if you really get 10 gram per liter out of your expression platform really depends on the combined effort and improvement of all these factors. And the plasmid factor can be an important part of that. There is not the CHO cell line. There is a whole zoo of different CHO cell lines, and even the CHO cell lines that have a name that are on the market are continuous. By every lab is using basically a different variant because of the genomic plasticity. Yeah, they continue to develop. But still, yeah, CHO cell lines, the most commonly used are, yeah, the ones mentioned here, CHO-K1S and CHO-S, and the DHFR knockout cell lines, DG44 and DUXB11, and some GS knockout cell lines, such as HD-BIOP3, CHO-K1SV, and Chosen. If you would be able to generate the ideal vector, what features should it have? Well, here's a vector wish list. The vector should be easy to introduce your gene of interest, so easy vector construction. Bicistronic vector would be ideal for expressing like light chain, heavy chain from the same vector. And it should be stably propagated in E. coli without mutations being generated. Selection after transfection into your cell line should be optimized. So it should be fast and easy with an optimized selection pressure, meaning that there's a balance between the how stringent you select and how easy it is to still get clones growing out of your selection procedure. There should be a high percentage of high expressing clones in you after the selection, after applying the selection pressure. And when you co-transfect two different vectors, each one should be expressed at a similar rate and not randomly distributed. And in the end, you want to isolate clones with superior clone performance. That means that you need high expression levels, having a strong promoter, but you don't want the expression to be epigenetically downregulated. The mRNA that is generated needs to be stable and efficiently translated, and the product that comes out needs to be of high quality. Vectors that are currently being used are still, many of them are still, well, they've basically been developed 25 years ago and not changed a lot since then. So many companies are still using very basic vectors with a standard CMV or EF1 alpha promoter. And especially CMV promoter is quite sensitive to epigenetic downregulation. Also, resulting in pools and containing clones in which in a variety of clones, and many of them have relatively low expression levels, and only a small fraction is really the high expression, high titers that you're looking for. If you co-transfect vectors, there's large variation in the relative expression between co-transfected vectors. So you really have to perform even large screening to make sure that both vectors are expressed at the expected ratio. And this shows that vector development hasn't received the attention it deserves. What I want to show you here is that a premium alternative is available, and that brings me to the introduction of the 2G Unic technology. So the 2G Unic vectors, they have been developed to improve multiple aspects in the protein expression pathway. So not just the epigenetic regulation, preventing that the expression is epigenetically downregulated over time. Not just a high transcription by introducing a stronger promoter, not just by improving mRNA stability or transport internally, and not just translation improvement by improving ribosome affinity, but really combining all these features in a single vector. So, and this is how we define 2G Unic technology as a combination of proprietary genetic enhancer elements fused for synergistic improvement of protein production level. And that means that it improves transcription, meaning more mRNA per copy of your per cassette that is integrated in the genome of your cell, and increased translation, mRNA stability and transport, and altogether resulting in more protein per produced mRNA molecule. And more mRNA per integrated copy and more protein per mRNA means that there is an increase in the expression of your protein per integrated copy in the genome. And that also affects your clone properties. So you will have more high-producing clones that are expressed at a stable level. This is how we apply the technology. So the 2G Unic element is composed of different elements. So my pointer. So there is a transcription enhancer element fused to human CMV promoter. In most cases, we have seen that human CMV promoter is performing really well in the 2G Unic constructs, but it can basically be another promoter as well. And there is a 5' UTR that we call the translation enhancer element, which is basically a 5' UTR with a intronic sequence. And this element can be fused directly to a gene of interest with a polyannulation signal. This is how it is applied in a vector. So basically we offer two options. The first option is if you have already a vector, maybe with some proprietary technology, your favorite selection marker, we can add the 2G Unic element as a single element into an existing vector. Second option is that we offer a complete vector system where you just need to introduce your gene of interest. And we have those vectors for single cassette or bicistronic ready for different selection markers. So yeah, let me tell something about how 2G Unic is used in cell line development programs. We mentioned an ideal vector should be easy to construct, at least in our proteonic vectors or complete vectors. We offer easy vector cloning through a Golden Gate assembly. That means that you just need to mix the vector with your genes of interest. So a light chain and heavy chain can be mixed together, maybe as synthetic fragments or already cloned in a plasmid. Just mix Golden Gate assembly, and you the resulting reaction, resulting plasmid will be the expression plasmid that can be transfected into your cell line. Also, if you want to use the 2G Unic element in your existing vector, then we just provide this element, and you can use that, introduce that into your existing vector for inserting a gene of interest, or Proteonic can do that for you. Then you want to have an efficient, stable pool generation protocol, or well, if you use stable bulk pools during the cell line development, of course. So what we see is that after transfection, basically the transfection the protocol does not need to be changed in order to use the proteonic vector. So the same protocol for transfection and selection can be used here, and that will result in a high percentage of high expressing cells. There's a small graph shown there comparing a reference sequence, a reference plasmid to a proteonic 2G Unic vector. And this is a typical improvement that we see because many more clones in your stable bulk pool will be high expressing. There's a significant increase of 2 to 6 folds typically for platform vectors. Will be shown in stable bulk pools. So, and of course, this should generate superior production clones. And there's another small graph shown underneath showing what it looks like. After the selection process, you end up with some top clones that are being tested in a fed batch process. And this is typically how this can look, showing again a platform vector next to a proteonic vector with significantly increased titers in the top clones. These titers are stable and quality of the protein is not affected by the higher titer. And the performance is scalable, so these clones can be taken, expanded into bioreactor and still give the same high performance. And some companies also use this to reduce the screening requirements because a larger fraction of the initial transfectants, the stable transfectants, is already high expressing. So a key aspect is that you don't need to change the cell line development process, basically. And we have seen that applying these the 2G Unic technology improves protein production across platforms and products. So we have seen similar improvements in CHO cells, CHO-K1, GG44, and different GS knockout cell lines, including CHO-K1SV, Chosen, and HT-Biop3. We have seen similar increases for different selection markers. So GS and DHFR as metabolic markers, but also antibiotic markers give similar results. Expression has been increased for different types of molecules, including antibodies, bispecifics, Fc fusion proteins, etc. And this is another way of showing that there is 2 to 3-fold increase or even higher at the clonal level for different cell lines and different molecules. So the increased performance with 2G Unic does not impact product or cell line features. So this is data that we have obtained from client studies. Typically, it's included in an evaluation study. Client always wants to confirm that cell line stability is not affected, and which they confirmed, that the process is scalable into a bioreactor, and that product quality is not affected. And also there we have never seen that product quality is affected. So the range of product quality variations that are typically seen for a standard or reference vector are also seen in the 2G Unic vector, meaning that there is still selection possible for clones that have the ideal product quality. Now I would like to show some examples, case studies. So if you have a standard antibody that is easily expressed, you can already, it's nice to have an increased expression, as can be seen here for an antibody molecule, and so the top graph is showing this for a bulk pool titer. being significantly increased between the platform vector and the 2G Unic vector. So this is basically the picture that I showed you a few slides ago, showing that titers in the clones are increased from around 1.5 gram per liter to around 4.5 gram per liter in the top clones. So a threefold increase was obtained in this system. But yeah, for more difficult to express clones, it could be it could have a higher value to increase titers. So and we have seen that proteonic the 2G Unic vectors have a very high success rate of also increasing titers for difficult to express proteins. And we've worked quite a lot also with the multi-specific antibodies and yeah, so the picture shown here is an example of a company we worked with. They already had quite a good vector, I must say, that gave quite good titers for more standard antibodies, but they struggled with their bi and tri-specific that had that were quite demanding and gave low expression levels. And we helped them to improve their vector performance by introducing our 2G Unic elements. And initially their titers went up from about 1 to 3 gram per liter. And they followed up doing more process development, exchanging some of signal peptides and other optimizations. And in the end, they reached titers over 10 gram per liter. So that's the example shown here for one of their bispecifics, but it got similar results for trispecific molecules. But a significant part of the well, I think there is a significant contribution of the 2G Unic technology, bringing the titers already from 1 to 3 gram per liter, so triple the titer there. This is another example of a difficult to express protein, a bispecific. Again, it's a study performed by CDMO. They had a client molecule that was expressed in the Horizon Discovery Bio3 cell line. And they had, so they compared the expression of a competitor vector sequence and the 2G Unic vector sequence. And again, the titers with the proteonic vector were much higher. So the top clones here from a large screening, most clones expressed below 1 gram per liter, whereas titers for the 2G Unic clones went up to 4.5 gram per liter, and also with high quality of bispecific production. So we've also worked with quite a few companies that already had a technology in place. So they had often had a proprietary technology, including chromatin modulator technologies such as UCO, SAR and MAR sequences, or transposon technology. And they were so they were achieving already quite good titers, but were interested in further increasing the titers. Another technology I want to mention is the recombinase mediated cassette exchange targeted integration, which typically is using single copy expression. And we have had also projects where companies wanted to increase titers from these platforms. And we have seen that this is quite successful, and all of these technologies larger pharma companies have taken licenses and apply it in their production system. So this is an example for an RMC cell line. This company was producing polyclonal antibodies through a library approach, meaning that in their vector construction process, they had a library that needed to be transformed into an expression vector library, several cloning steps that were involved to generate this vector library. And in the end, it was transfected into cells to generate stable pools, producing the polyclonal antibody. Quite demanding on the side of stability, also already on the E. coli vector cloning side. That was successful already, so that was compatible with 2G Unic elements. And also the 2G Unic vector helped them to achieve way better titers from their polyclonal pools. So going up from less than half gram per liter to almost 2 gram per liter in a fed batch process. And I think that is quite a good performance for a targeted integration single copy system. What we have seen in several of these projects is that in many cases, the 2G Unic elements are able to increase the performance in those vectors, but it depends on the integration site in the genome. So it seems that for some of these sites, there's not a large benefit of using the 2G Unic, whereas in other sites, the effect can be quite good, like shown here in this example. Transposon technology, also a technology that is being used more often these days and is giving quite good titers in many cases. Not all transposon technologies are as good as others, so there's also quite a bit of variation between the technologies. But we've seen that the 2G Unic elements are nicely compatible with those transposon vectors and transposases. So the picture on the left showing a comparison that a client did. So these are the pool level titers and the clone titers, the top clone titers for two different transposases compared to the 2G Unic vectors in a random integration and showing that in this case the performance of the 2G Unic vectors was better than the transposases. It's not always the case, so we have seen that, for instance, the super piggyback transposon is performing very similar to the 2G Unic vectors, giving way higher titers than the standard platform, like threefold higher compared to platform vectors. We have seen that in different cell lines, including CHO-K1SV and HE-Bio3 cells. But what we've also seen is that if you combine the two technologies, so add the 2G Unic elements to the transposon vectors, that there's an additional increase in titers is possible in the generated cell lines. So 2G Unic improves additional factors that are not currently covered by the transposon technology, which makes sense because the elements were really designed to improve multiple factors, such as including the translation enhancing features that are not present in the standard transposon technology vectors. So this is to my final slide to summarize 2G Unic technology. I think I've shown here that the technology can really boost recombinant protein production, providing transcription and translation enhancement, resulting typically in a 2 to 6-fold boost of expression. To use it, you don't need to change a lot in your process, and also the protein stability, product quality are maintained. So technology is optimized for mammalian cell lines, and really works well with many difficult to express complex proteins, with quite a high success rate. Also for the difficult to express proteins. It's already being used by a few of the top 20 pharma and biotech companies. And currently there are cell lines in phase 2 and beyond. Then, one more slide. This is so we also have we're using currently these 2G Unic elements and variants thereof to increase expression, and also in viral systems, cell and gene therapy vector systems, including lentivirus and AAV, and we have some good initial results showing that we can increase viral titers, mostly on the lentiviral side, and also payload expression in AAVs. And we have seen that it's, for example, we can combine the 2G Unic elements or versions thereof to increase expression from tissue-specific promoters, for example, in AAV virus particles. So this is ongoing work. Just I wanted to mention that technology can be applied broader than just in the CHO cell line development. This is really the final slide. So yeah, over to Jonathan.
Excellent. Well, thank you, Maurice. That was great. And yes, if there's any questions, please feel free to type it into the question box, and I will have a look to see what we have. Here's a nice, easy one, I think, to come in. So the question was, what is the size of 2G Unic?
The size of 2G Unic, it depends on the promoter that is being used. For the transcription enhancer element is around 1.5 kb. So that's added upstream of the CMV promoter. The 5' UTR element is a little under 300 base pairs. So the construct we typically use, including the human CMV promoter, is around 2.7 kb.
So, and there's a question that's actually come in via the chat as well. Maybe we'll do that one. So it's, how does the copy number of your vector compare to reference vectors?
Very good question. We have seen that it doesn't really change copy numbers. What we have seen a few times also in client studies was that the copy numbers in the top performing clones were a little bit lower than with 2G Unic, were a little bit lower compared to reference. I think it's something that you see a lot in cell line development projects, that the top producing clones have relatively low copy numbers, I would say. And it seems to be that if production per copy is higher, then the top clones can even have lower copy numbers and still produce way higher titers compared to the reference vectors.
And another one that's coming is, so in addition to question, sorry, as I publish it, this appears from my screen. In addition to CMV promoter, what other promoters work well with the 2G Unic system?
Many promoters work with the 2G Unic. The system of the I didn't really go into that, but the mechanism of the 2G Unic is basically an intronic promoter, where the enhancer elements, the transcription enhancer element upstream of CMV is also actively participating in transcription, and the CMV is basically present in an intron that is being spliced out. We have seen that some of the promoters, and that's mostly mammalian promoters, are not correctly spliced out or can have some artifacts there, and typically viral promoters work better than mammalian promoters.
The next question is any limitations on the gene of interest size or the number of chains that can be incorporated into a single vector?
Not really. We have been using some quite large genes for some projects. So no, there's not really a limitation. So what can be done, for example, is making very large plasmids to introduce into by targeted integration containing multiple genes on the same plasmids. So it will affect your transfection efficiency, but if you only have a single copy, it doesn't really matter. So there is no limitation on the vector side. The efficiency will depend on the system, on the platform that you're using.
And it could be a bit of a follow-up as well. So how does it work with multiple vectors for complex targets like bispecifics or trispecifics?
Yeah, that's also a very good question. And what we have seen is that because the expression is basically independent from the integration site, you see that the expression levels are also more relative to the ratio of how you mix the plasmids. So if you mix two plasmids in a transfection mixture, they will be expressed at more or less equal ratios. So for example, well, it works really well for the multispecifics because if you typically need more than one vector, if you have one vector expressing one heavy chain, light chain, another vector having another heavy chain, light chain, both you will quite easily find clones that express both the chain combinations at equal ratio. So that's one of the reasons why the proteonic vectors work so well with the more difficult to express multispecifics.
There's so what percentage of clones are stable and what criteria is used for defining stability?
Yeah, so stability was typically defined by the client who did the tests. So someone less than 20% decrease in expression after 60 generations. For others, it's like 80 generations. So it really depends on the clients. Typically, they still found clones that were non-stable, but it was very similar to their to their reference system. So it's not really that you increase the percentage of clones that are stable. It more depends on the system, on the platform that you're using.
I was on mute.
Yeah.
Sorry, what's next from Proteonic? Any plans to incorporate transposase technology?
We don't have our own proprietary transposase technology, so we're compatible with transposases. So as long as the company offering the transposase is allowing you to make the combination with the 2G Unic elements, then we can provide that element.
So how do we test the technology in our workflow?
Yeah, so typically for evaluation studies, we see that companies choose to use like an easy antibody and a more difficult to express protein, maybe another format of a difficult to express protein. So 2 to 3 proteins, and then compare their existing vector with a 2G Unic vector, and if it's worth to also make the combination of 2G Unic with the existing platform, that would be a third vector that could be included in an evaluation study. And in that case, Proteonic can do the vector designs, provide the vectors, including your gene of interest, if you can share that sequence, provide that back to you, transfection ready for the evaluation study.
I think that's the questions so far. If anyone else has any last question they want to put in, quickly type away. Otherwise, I think it's probably time to sort of thank everyone for joining us for the webinar today. I hope it's been useful and informative. Please feel free to reach out to either myself or Maurice if you want to learn more about the technology, or you have any questions at all. I'm liking someone's clapping, so that's good. I appreciate that, the claps. And hopefully we'll see you at future conferences in actual in real life versus just virtually. So appreciate everyone's time today, and look forward to engaging with you in the future. And thank you again, Maurice. That was great. Cheers, everyone.
Thank you, everyone.